Okay. Good evening, everyone. So today we will start with the microbial genetics. It's very interesting and a little complicated at the same time, but I will try to make it as easy as possible. Um, just let me know whether my screen is visible to you and my voice is audible or not.
Yes, both are clear.
Okay. Okay. Is the PPT visible?
Yes,
Okay. So microbial genetics. So we are humans, right? And we do have a chromosome. And what is there in the chromosome? Chromosome is made up of DNA, right?
yes,
Huh. So similarly, microbial organism, we will focus mostly on a bacteria. Okay, it is the most prevalent microorganisms that that we have in our surroundings. So we'll take an example of an E. coli and try to understand how the microbial genetics works. Okay, so let's start. So genotype and phenotype. The genotype of an organism. What is genotype? So genotype of an organism is a genetic makeup, the information that codes for all the particular characteristics of the organism. So we all have our genes, right? So some of us are fair, some of us are dark skinned, some of us have curly hair, some of us have straight hairs, some of us have brown eyeballs, some of us has green eyes. So what does these things depends on? This thing, all of these things are dependent on the genes. Okay, so the genes comprises of the genotype, and it is not always the case that if you are having a gene for a certain disease, okay, it will always be expressed in your lifetime. It may be expressed in your progeny. Understood what I am saying? It may be expressed in your progeny. आपके बच्चों पे ये आ सकता है या फिर ग्रैंड चिल्ड्रन में भी वो डिजीज आ सकता है. So genotype is not always, this does not always results in a same phenotype. Understood? Now we will come to phenotype. Phenotype refers to the actually expressed properties. ठीक है, कि किसी का आँख का कलर ग्रीन है जो उसका जीन में एनकोड था और उस जीनोटाइप के लिए ये फिनोटाइप पॉसिबल हुआ है. So phenotype is the actual expression. Understood? It is the manifestation of the genotype. I hope it is clear. Okay.
yes,
Okay. Now the molecules of genetics. So जिन लोगों ने मेरा बायोमोलिक्यूल्स का क्लासेस किया है उनके उनको ये बहुत क्लियरली आना चाहिए बिकॉज आई हैव टेक आई हैव taken numerous lectures on biomolecules. Like the main portion of the syllabus was biomolecules that I was supposed to take. Okay. The main molecules of the genetics are called nucleic acids, right? What they are called? Nucleic acids. What are there in nucleic acids? Deoxyribonucleic acids and ribonucleic acids. Okay. Shubham, can you tell me why DNA is considered more stable than RNA? If you don't know, you can just say that you don't know.
but,
Okay.
mm,
So can you spot any difference? Have you done the classes of my like gone through the lectures? No, right?
না সবকটা দেখা হয়নি।
Okay. So DNA and RNA differ in a hydroxyl group. RNA has a hydroxyl group at the 2' position of the pentose sugar. 1' then 2'. So that sugar can very well form that OH bond can very well form hydrogen bonds, and also that oxygen can perform various nucleophilic substitution reactions in the other groups of the RNA. thus making it very much unstable. We will never want our DNA to interact with other biomolecules. Okay, it will interact with only the desired biomolecules, that are the various type of proteins that function to replicate it, right? Okay. So all the genetic information are stored as a sequence of bases through nucleic acids, mainly in DNA, RNA, and in okay. So DNA is the main genetic information storehouse, but in certain viruses, they also have RNA genome, which is only seen in viruses. Okay, so you can see a basic structure of a nucleotide. What is a nucleotide? Nucleotide composes of a phosphate group, the pentose sugar, and the nitrogenous base. Okay, Shubham, can you tell me how many nucleotides are there, or how many nitrogenous bases are there?
Nitrogenous base ATGC,
ATGC. So what are ATGC?
adenine, thymine, guanine, cytosine.
Very well, very good. Okay, so DNA serves as the organic genetic material. ठीक है? It is the organism genetic material. It is divided into two functional unit. It is divided into functional units, that are the genes. Okay. Do you think all of your DNA codes for genes? No. There are certain regions of the DNA which doesn't codes for any particular kind of proteins. ठीक है? But they are still present. Okay. So most of the DNAs are double stranded. The two strands are held together by hydrogen bonds between A and T, or G and C. It consists of non-identical complementary base sequences. Understood? Non-identical. That is, no two DNA of organism we can say that they are exactly similar to each other until and unless they are inbreeded. Okay, in inbreeded also there are certain cases where we can see genetic differences. So what is inbreeding? So breeding within the offsprings. Okay, complementary
Hmm.
base sequences. We all know that adenosine complements with thymidine and guanosine complements with cytosine, right? Now RNA, that is the ribonucleic acid, it is structurally similar to DNA, except most RNAs are single-stranded and sugar is ribose instead of deoxyribose. Understood? In DNA we have deoxy, and in RNA we have only ribose. Okay. And what is the other difference? We have uracil in place
US.
of thymine.
Hmm.
Okay. Okay. See, a prokaryotic chromosome. So I don't know how much this is visible to you. I will try to. Huh. So can you see this tangled mass of looping strands of DNA? This is nothing but the DNA. Okay. It is emerging from a disrupted E. coli cell. Disrupted means its cell membrane is destroyed. Okay. And they're all part of a single chromosome. So see how much it is packed. Okay. Understood? This is under a electron microscope.
Hmm.
So these are packed in such a way that we can only see a small dot in the cells, right?
Huh.
Ah, okay. This diagram on the right hand side, it is showing a genetic map of E. coli chromosome. So you don't need to remember this, but this just to show that it can be mapped like this. Okay. Okay. Now bacterial chromosome. So bacteria typically have a single circular chromosome. We all know that bacterial chromosomes are single circular chromosome consisting of a single circular molecule of DNA with associated proteins. Understood?
Hmm.
So we all know that DNA, the nucleic acid, is always associated with certain proteins which are essential for its survival of the nucleic acid. The chromosome is looped and folded and attached at one or several points to the plasma membrane. Okay, so it is a very new thing. You will ask that, Bhaiya, we haven't, Dada, we did do that. It is located in the center of the bacteria, but is it really attached or several points to the plasma membrane? Yes, it is. ठीक है, the DNA of E. coli, the most studied bacterial species, has about 4.6 million base pairs. Okay, see how many? 4.6 million base pairs and is about 1 millimeter. ठीक है, 1 millimeter, 1,000 times longer than the entire cell.
10 NG টা কি?
It is nanograms.
আচ্ছা আচ্ছা।
ठीक है. However, the chromosome takes up only about 10% of the cell's volume. Okay, but এটা এত লম্বা, but it still only takes only 10% of the cell's volumes because the DNA is twisted or supercoiled, much like a telephone cord when you put the headset back on the receiver. So পুরোনো দিনের telephone, if you have seen old day telephones, they have coiled loops. Okay, what they have coiled loops. Understood? So the
হুম
DNA
হুম হুম।
somewhat like looks like that. The location of the genes on bacterial chromosomes can be determined by experiments on the transfer of genes from one cell to the another. So we will study this type of experiments in this lecture. Okay. In recent years, the complete base sequence of several bacterial chromosomes have been determined. So scientists, what they have done, they have determined a complete list, made a complete list of various bacterial chromosomes. Hmm. Computers are used to search for the open reading frames, that is the regions of DNA that are likely to encode a protein. तो मैंने अभी क्या बोला था, पूरा डीएनए कभी भी कुछ के लिए एनकोड नहीं करेगा, उसमें से भी कुछ कुछ रीजन्स एनकोड करेगा. तो ऐसे रीजन्स को बोला जाता है ओपन रीडिंग फ्रेम. Okay, these are actually base sequences between start and stop codons. So any idea about what are codons, Shubham?
ওই জেনেটিক কোড যেগুলি
Okay,
থাকে।
what are codons?
ডেফিনেশনটা বলতে পারছি না এই সময়।
Okay, ठीक है, you know it, that's good. The sequencing and molecular characterization of genomes is called genomics. Understood? What is genomics? The
হুম হুম হুম।
sequencing and molecular characterization. So what is molecular characterization? Doing PCR to understand the structure, to understand the sequences. Okay, so these are all the molecular characterization. Okay, can you read this difference for me?
হ্যাঁ, difference between prokaryotic and eukaryotic chromosome. Prokaryotic versus eukaryotic chromosomes. Prokaryotic chromosomes. Many prokaryotic contains a single circular chromosome. Other side, eukaryotic contain multiple linear chromosomes. Prokaryotic chromosomes are condensed in the nucleoid via DNA supercoiling
Hmm.
and the binding of various architectural proteins. On the other side, eukaryotic chromosomes are condensed in a membrane bound nucleus via histones. Because prokaryotic DNA can interact with the cytoplasm, transcription and translation occur simultaneously.
Hmm.
Other side, in eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. Most prokaryotics contain only one copy of each gene, such that they are haploid. On the other side, most eukaryotes contain two copies of each gene, such that they are diploid. Uh, Non essential prokaryotic genes are commonly encoded on extrachromosomal plasmids. Some eukaryotic genomes are organized into operons, but most are not. Prokaryotic genomes are efficient and compact, containing little repetitive DNA. Extra chromosomal plasmids are not commonly present in eukaryotes. Eukaryotes contain large amounts of non-coding and repetitive DNA.
Okay. So the things that we already know, that prokaryotics contain a single circular chromosome, but eukaryotics contains multiple linear chromosomes. Okay. Okay. So prokaryotic chromosomes are condensed in a nucleoid via DNA supercoiling, but they do not have a nuclear membrane, right? They are not membrane bound. This is a very major difference. Whereas eukaryotic chromosomes are membrane bound and they are bound to proteins that are called histone proteins. Very important. Okay. Because prokaryotic DNA can interact with the cytoplasm, transcription and translation they occur simultaneously. Okay. In eukaryotes, transcription occurs in the nucleus and the translation occurs in the cytoplasm. We all know this, right? Most prokaryotes contain only one copy of each gene. There is only one copy of each gene. So there is always a chance of gene loss. That actually sometimes happens in bacteria. There is loss of essential genes and the bacteria eventually dies. It is also a target of future, like targeted therapy to work against the AMR, antimicrobial resistance. Okay, if by some means you can force the bacteria to lose its genes, then the bacteria will die eventually, naturally. Okay, without the use of any antibiotic. This is a research gap. Okay, understand very well. If you are targeting to study antimicrobial resistance, you can go to this topic. Okay, most eukaryotes contain two copies of genes. We all know that we are diploid organisms, right? Non-essential prokaryotic genes are commonly encoded on the extra chromosomal plasmids. Okay, can you name one very common extra chromosomal plasmid? अच्छा बोल ही दिए थे. Extra chromosomal nucleic acid, that is plasmids. Plasmids are also very important in the life of a bacteria. Okay, extra chromosomal plasmids are not commonly present in eukaryotes. In eukaryotes, extra chromosomal plasmid is not present. Understood? Prokaryotic genomes are efficient and compact, containing little repetitive DNA. प्रोकैरियोटिक जीन्स में रिपीटेटिव डीएनए नहीं होता है, बहुत ही कम अमाउंट में मिलता है, बट यूकैरियोट्स कंटेन लार्ज अमाउंट्स ऑफ नॉन कोडिंग एंड रिपीटेटिव डीएनए. एक ही चीज का बहुत सारा रिपीटेशन होता है, ताकि अगर कोई कुछ रीजन से कट भी हो जाए एनी जीन, ठीक है, तब भी we can always survive. So there are certain kind of viruses, they can insert their gene into our chromosome with the help of certain proteins called integrase protein or integrase enzyme. But since we have multiple copies of the same protein inside one molecule of the DNA, that's why we survive, we do not die. Understood?
এই জায়গাটা একটু রিপিট করো, বুঝতে পারলাম না।
ওকে, প্রোক্যারিওটটা বুঝতে পেরেছিস?
হ্যাঁ।
আচ্ছা, ইউক্যারিওটসে কি হয়? এইচআইভি নাম শুনেছিস? এইচআইভি, হার্পিস,
হুম,
ঠিক
হুম।
আছে, হার্পিস
শুনি।
সিমপ্লেক্স। হ্যাঁ, তো এদের মধ্যে একটা এনজাইম থাকে, এটাকে বলা হয় ইন্টিগ্রেজ এনজাইম। ঠিক আছে, তো
হুম।
এই এনজাইমটা কি করে, এইচআইভির যে জিনোমটা থাকে সেই জিনোমটাকে আমাদের ডিএনএর মধ্যে ইন্টিগ্রেট করে দেয়। ঠিক আছে, মানে ওই ডিএনএটাকে আমাদের ডিএনএ ঢুকিয়ে দেয়, লাইক কাট করে
আচ্ছা,
পেস্ট করে দেয়, সোজা কথা।
আচ্ছা, আচ্ছা, আচ্ছা।
ঠিক আছে? তাতে যদি আমার যেই জায়গাটাতে ঢোকাচ্ছে, ঠিক আছে? সেখানটা যদি ওই প্রোটিনের যদি আর কোনো কপি না থাকে, তাহলে ওই প্রোটিনটা তো আর সিনথেসাইজ হবে না। হবে কি?
না,
না, হবে
না,
না।
না।
তাই না?
হুম,
বাট যেহেতু আমাদের জিনে অনেকগুলো কপি থাকে সেম প্রোটিনের, সেজন্য আমাদের অতটা এফেক্ট পড়ে না, উই সার্ভাইভ। বুঝতে পেরেছিস?
তা সে মানে ওই কপিটা কি মানে প্রয়োজন নাকি মানে দরকার নেই সেই মানে
না,
কপিটা থাকাটা?
অফকোর্স এই এই ক্ষেত্রে প্রয়োজন। ঠিক আছে, যখন একটা ভাইরাস অ্যাটাক করছে, নিজের ডিএনএটাকে ইনকর্পোরেট করে দিচ্ছে, ঠিক আছে, যদি আর কপি না থাকতো তাহলে সেই প্রোটিনটা সিনথেসাইজ হতো না।
উম, সেই পার্টিকুলার সেই প্রোটিনটার সিনথেসাইজ হওয়াটা কি প্রয়োজন পড়ছে সেই পার্টিকুলারটা
ওকে,
যেখান থেকে
ওকে। যদি ধর ওটা কোনো এসেনশিয়াল প্রোটিন হয় বা হাউসকিপিং প্রোটিনের জায়গা, হাউসকিপিং প্রোটিন মানে জানিস?
বলতে?
হাউসকিপিং প্রোটিন মানে যাদের অ্যামাউন্ট আমাদের বডিতে সবসময় দরকার, যেমন এটিপেজ।
আচ্ছা আচ্ছা
ঠিক
আচ্ছা।
আছে,
হুম
এটিপেজ ছাড়া উই আর ডেড। এটিপেজ বুঝলে না, এটিপি সিনথেসে, এটিপি সিনথেজ, এইগুলো,
হুম হুম।
এগুলোকে হাউসকিপিং জিনস বলে। এগুলো সব সময় প্রোডাকশন হতেই থাকে। ঠিক আছে, তো ভাইরাসটা যদি এর কোডিং সিকোয়েন্সে এসে ইনসার্ট করে দেয় নিজের ডিএনএটাকে, তাহলে তো গেল এই প্রোটিনটা, মিউটেশন হয়ে গেল। ঠিক আছে।
তাহলে মানে এইটা মানে পার্টিকুলার কিছু প্রোটিনস এর ক্ষেত্রে সেটা মানে আর কি হচ্ছে, সব প্রোটিনস এর ক্ষেত্রে নয়।
সব, যে কোনো প্রোটিনের ক্ষেত্রে হতে পারে।
আচ্ছা
ঠিক আছে, যে কোনো
আচ্ছা
প্রোটিনের ক্ষেত্রে হতে পারে। হুম।
আচ্ছা আচ্ছা।
ওকে, নাও লেভেলস অফ জিন রেগুলেশন। কি কি লেভেল, হোয়াট আর দ্য লেভেলস অফ জিন রেগুলেশন? ওকে, অ্যাজ ফার আই নো এই বছরের টিআইএফআর এ এরকম একটা প্রশ্ন এসেছিল, which of the following has not been discovered yet বলে ডিএনএ আর প্রোটিনের কিছু একটা দিয়েছিল। হুম। The expression of a gene into functional proteins can be regulated at multiple levels. Transcription, that is regulation of rate at which the gene is transcribed. Transcribed, our DNA is transcribed into mRNA, right? mRNA transcript stability, it is the half-life of the transcripts, and after an mRNA is produced, it needs to be converted into a functional protein. That is translation, regulation of translation of mRNA, very important. Next comes the post-translational modifications. It is important because otherwise proteins are very much prone to be cleaved by polypeptides. Okay, addition of chemical groups. Okay, these are very important. Okay,
হুম হুম হুম।
so DNA replication will always begin at the origin. So this is a TEM, that is the transmission electron microscope resolution image of a replication fork of a bacteria. See, this is a duplicate diagram. Okay, replication fork A, we all know that replication proceeds of the same genome. What is replication? The replication of the same DNA
ডুপ্লিকেশন।
duplication, right? So it begins at the origin of the replication. So origin replication is start হচ্ছে, then there is a formation of a replication fork. There is a parental strand and the daughter strand. Okay, and there is a, so in the replication fork we know that two strands are separated, right? Two strands are separated.
হুম
Okay, so this is not a replication fork, understood? This is a entangled chromosome, and this particular region is a replication fork, understood? And it is known as replication bubble. It is known as replication bubble where the replication is taking place. Okay, so the whole genome will be copied and there will be termination of the replication, and from there we will get two separate new chromosomes, two copies. Okay, a mutation. Now, what is mutation? A mutation is any change of the sequence, change of one nucleotide to another. এটা একভাবে হতে পারে, ওয়ান নিউক্লিওটাইড থেকে একটা নিউক্লিওটাইড হলো। দেয়ার ক্যান বি ইনসারশন, একটা নিউক্লিওটাইড ইনসার্ট হতে পারে, আবার ডিলিট হয়ে যেতে পারে। ঠিক আছে, দেয়ার ক্যান বি ইনভার্সন অর রিকম্বিনেশন অফ ডিএনএ ফ্র্যাগমেন্টস। In next class, we will focus mostly on the recombination of the DNA fragments. Okay. Now,
হুম।
what causes these mutations? Errors in DNA replication, We all know that we have a DNA polymerase which has an error rate, and we also know that there is a DNA repair mechanism. Okay. Now next will come the chemical mutagenesis. Due to certain chemicals, there is mutation. Now high energy electromagnetic radiation can also cause DNA damage. UV light, X-ray, gamma rays. Okay, what are gamma rays? They are rated by radioactive substances. They can all cause DNA mutations to certain extent. Okay. So mechanism of gene transfer. So there are two very well-known theories: vertical gene transfer, that is via binary fission, that is from mother to offspring. अंडरस्टूड? एक सेल है जो दो सेल में डिवाइड होके अपना जीन को डुप्लीकेट करके आगे बढ़ रहा है. बाइनरी फिशन, वी हैव ऑल स्टडीड दिस इन आवर जूनियर क्लासेस. बाइनरी फिशन. ओके. नेक्स्ट होता है हॉराइजेंटल जीन ट्रांसफर. हॉराइजेंटल मतलब एक ही जनरेशन का दो सेल हो सकता है, मतलब साइटोप्लाज्मिक डिवीजन नहीं हो रहा है सेल का, बट वाया वेरियस मेथड्स. We will learn that methods: conjugation, transformation, and transduction. So the two bacterial cells can link to each other and can transfer or share one strand of a particular region of the DNA with another bacteria. Understood? Transfer within the same generation.
Mhm.
Okay, so what is homologous recombination? Let's see. So consider this is a DNA fragment from donor cell. Okay, so we know that in recombinant technology, we can force a bacteria to uptake certain kinds of genetic information, right?
Mhm.
So it can be any donor cell. It is not necessary that it will always have to be another bacterial cell. It can be any donor nuclei, or donor DNA. This is the chromosomal DNA. Just follow the sequence. Okay, A B C D capital, and here it is A B C D in small letters. And see, it is in the 3 prime of this strand. Okay, now the it is entering the bacterial cell. Okay, then the donor DNA aligns with complementary bases. So there will be a region where complementarity will be there with the bacteria. Okay, so
Mhm.
see, only this much region is complementary. Can you see? Till A region, the capital A. Understood? Capital A and small a are complementary. So hence they have combined with each other. Okay, but small b, small c, small d, they haven't. Okay, so there will be recombination between the capital A and the small a region. Okay, and the other regions will be degraded. The capital A will move to the donor DNA, and the donor small a will move to the bacterial chromosome. So this is called homologous recombination. Okay, so but the rest of the DNA will be degraded. Why it will be degraded? Can you tell?
Degraded.
Degraded means it will be cut into small pieces and it will be taken as a waste, considered as a waste. This is because this is a DNA fragment, not a complete DNA, and it is not having its own self-replication mechanism. But if it was a plasmid, what is plasmid? Plasmid is a self-replicating extra chromosomal DNA, right?
Mhm.
Uh, so if if this was a circular plasmid or it was having an origin of replication, then this DNA could have survived. ठीक है? So unless transferred DNA is circular with ORI, ठीक है? तो origin is ORI is origin of replication. ठीक है?
Mhm.
It must recombine with host DNA to be retained. So अभी मैंने जो retain, तो अभी जो मैंने बोला वही लिखा है कि unless it is having a origin of replication, otherwise it will die without being recombinant. Okay? So recombination can occur between homologous or similar DNA sequences. That is DNA with the same genes. That is complementary sequences. O- Okay, they are facilitated by special proteins, but the original DNA is always lost in homologous recombination. Understood? Okay. Okay, so mechanism of gene transfer. Bacteria can acquire DNA by three ways. Transformation, that is uptake and retention of external DNA molecules. It will uptake and it will retain the DNA molecules. What is conjugation? There is a direct transfer from one bacteria to another, and transduction, transfer of DNA by a virus. In conjugation, two bacteria will be linked to each other via conjugation tube. Okay. In transformation, there is no physical linking between two bacteria. In transduction also, there is no physical linking between two bacteria. Okay. Okay. Now let's see what is transformation. Okay. Transformation is very similar to what I have already explained in the uptake of the bacteria. Okay, we will go through this transformation, transduction, and Griffith's experiments in next class. I don't want to start this today. I know it is a heavy thing for you guys. Okay, just go through the previous things that you have learned in your class 11, 12. Griffith's experiment was there, right, in class 11 or 12. Okay, go through them. Next class, we will start with transformation and continue then forth. Okay. Any doubt in today's class? I hope this class was little interesting than previous plant biology classes. Okay. So I will end the recording 1 minute.